Given down below are some of the biogroups and pathways we found overrepresented in our meta-analysis. These are the databases that Correlation Engine is using to define these biogroups. And this is a filterable list. So say I only want to look at genes associated with pathways. I could click on Gene Ontology and say canonical pathways from the Broad Institute, and that will pull out all of those biogroups. Or maybe I'm more interested in finding overrepresented targets for transcription factors. I can click on this regulatory motifs and basically search all our databases just for that. We can also look for microarray targets, protein families, etc. We can take a closer look at some of these biogroups. The number one biogroup identified or most significant in our meta-analysis was vascular development. If you remember back when I talked about gave the introductory lecture to kidney cancer in part 1, I talked about one of the few forms of therapy they actually have for kidney cancer is anti-angiogenesis therapy, or preventing the formation of blood vessels to the developing tumor. Based on some of these biogroups, that looks like a pretty good idea. Obviously, there's not just one gene associated with this biogroup. There's many. Given here is a representation of all those genes in each individual comparison that were differentially expressed. If that gene went down compared to the control, it's represented as green, and those that went up are represented as red. And as you can see in the severe compared to the less severe forms of this cancer, there tends to be an increase in these type of genes. We can click on this to get more information. Again, here are all our comparisons that we use, 17. Here are the p-value, the significance of that finding, and here are all the genes in our gene list in each one of these comparisons that had this particular trend. So we can actually click on this top one. This is the comparison we used from the TCGA database. Click on that. So in this comparison to kidney renal papillary cell carcinoma compared to normal kidney, we found 11,569 genes differentially expressed. Again, the criteria is a p-value less than 0.05 and an absolute fold change greater than 1.2. Within this list, these are the genes that went up compared to the control, and these are the genes that went down compared to the control. Here is our biogroup, our vascular development biogroup, and as I mentioned, these are based on what the experts say about that group and the database that you're using. I believe this was Gene Ontology, but the experts, as far as human goes, are looking at 401 genes associated with this particular biogroup. When we compare these two gene lists, we find 246 genes in common. From those, 103 genes went up, and 143 of these genes went down compared to the control. Based on this result, the odds of this happening by random chance is 3.3 times 10 to the negative 50th. Now, Correlation Engine actually, it's not just a straight up Fisher's exact test. Hopefully there'll be a link that pops up right now where you can actually see the paper where they derive their statistics from. Some of this actually has to do with ranking, so that some of these vascular development genes are ranked higher than other genes. That means they have greater fold change values. So, and that figures out into the calculation of the p-value. But I'll let you read all up on the mathematics on it. Click out of here. We can take a look at some of these other biogroups. Again, we're looking at cancer. Genes involved in cell cycle seem to be, for the vast majority, seem to be going up, which makes sense. We have organic acid catabolic processes. I mentioned that with type 2, you typically see a mutation or sometimes frequently see a mutation in fumarate hydratase. This is a metabolic gene. So maybe this is an example of that metabolic shift that occurs in some types of cancers. It's called the Warburg effect, where it's shifting its glycolysis to produce more ATP to promote more cell growth. Apical part of the cell, genes involved with cell cycle. What I also find interesting, and you see this in a lot of cancers, is that these cells start out very differentiated, and when they turn cancerous, they almost go primordial. So they start losing those things that, differentiate it from the other cell types. So what you're seeing here, and probably in some of these, urological system development, kidney development, is you're seeing those genes that make a kidney cell disappear because the cancer cell doesn't need it anymore. I love this page because I think it gives you a good idea on what you should expect out of your meta-analysis. So I love to kind of come here, kind of take a look at the database and, see what I should expect. But what we're going to want to do, and our goal for this, is to basically take our genes, the genes in the gene list, the gene results here, and download them so we can analyze them with another program. So what we're going to do in the next video is basically take all of these genes and export them as an Excel file. Once we've exported them, now we can then upload them into another program that we're going to use called Partech Genomics Suite, which is a statistical graphical program. What we can now do is look at the different trends in these different genes across all these different data sets and basically cluster them based on the paths that they take across these different data sets. So that's what we're going to do. Before I do that, though, I want to show you one thing. Obviously, every time you log back into Correlation Engine, if you wanted to do the same meta-analysis, you wouldn't want to basically build it from scratch. So what you can do is go to this tab over here called Bookmarks. I click on that. And as you can see, I have now bookmarked this page. So all I have to do whenever I log into my Correlation Engine to retrieve my meta-analysis, all I do is click on this little link and I am back to my meta-analysis. This concludes the part 2 of our meta-analysis series. Like I mentioned, in our next series, what we will be doing is downloading all of this information, exporting it to another program, and doing further analysis, statistical analysis with Partech Genomics Suite. Until next time, thank you very much.